So we've talked about the different parts. Like I said, I've kind of been feeding this information to you piecemeal. And so today I'm hoping that we're going to be able to draw all those different parts, components of the immune system together so you can kind of get a broader understanding. All right. So we are talking about the adaptive immune system. And the adaptive immune system is made up of the lymphocytes. Okay. So that's the T lymphocytes and the B lymphocytes. All right, and we know that there's two types of T lymphocytes. There's the helper T lymphocyte, or the CD4 cell, all right, and then we have the cytotoxic T cell, which is the CD8 cell. Okay, and last class I talked about how for these cells, all right, to dock or bind onto an infected cell or a cell that's expressing an APC cell, an antigen-presenting cell, all right, they have to, if they're going to try to bind with that, all right, we have these specialized transmembrane proteins that the unhealthy or infected cell has or the APC cell has, and basically that's how these cells express the antigens on their plasma membrane. And we call those transmembrane proteins the MHC class 1 or class 2. Remember that? The histocompatibility complex, okay? And so it's this protein that would take the antigen and wear it on the end of it like a hat, okay? And then when the lymphocyte came by, it would see, all right, the antigen being expressed, and then it would examine it, okay? So now we're going to talk about what happens during that examination process, and now you'll hear me say terms like, these cells can initiate the immune response. Now we're going to start to talk about that, the actual immune response. So we're going to start off by talking about, okay, the activation of the helper T cells. Okay, helper T cells are also known as the CD4 cells. All right, remember the rule of 8s. CD4 cells, all right, are going to bind with the MHC class 2 molecules. Okay, 4 times 2 is 8. Okay. So in this situation, and if you remember from last class, the only type of cell that expresses an MHC class 2, all right, protein is going to be APCs, antigen-presenting cells. And there's 3 that we know of. We have our dendritic cell, the macrophage, and believe it or not, the B lymphocyte is going to be, and we're going to talk about its role as an APC today. Okay, so that's the third one that we're going to be discussing later on. Okay, but keep in mind, all right, so the T helper cells, the CD4 cells, all right, are going to bind with, all right, MHC class 2 molecules, which are present on our APCs. Okay, so when we talk about the APC presents an exogenous antigen, that's usually a foreign antigen. Okay, endogenous is from the inside. So the endogenous antigens would be, remember that first, oops, going the wrong way, guys. Remember this picture I showed you, the formation of the MHC class 1 molecule in a healthy cell? This is what your cells are doing right now when you're fine and dandy, okay? They're just expressing the self or endogenous antigen on their cell surface. So your immune cells, when they're coming through and doing their immune surveillance, they're like, okay, that cell's cool. It can be here. We're not going to do anything to it, okay? So notice that's class 2. Well, when this cell becomes sick, all right, when it becomes unhealthy and it expresses the foreign antigen or the exogenous antigen, all right, being class 1, it can only bind with the CD8 or the cytotoxic T cell. We'll talk about that, okay? So here we're going to be dealing with this scenario, okay? We have an antigen-presenting cell that's going to be expressing that MHC class 2 molecule, right, that it got from a virus or bacteria, all right, and it ate it up through phagocytosis, it digested it, broke it up, and then as it was developing its MHC class 2, all right, proteins, it threw on that foreign protein, and now it's expressing those exogenous proteins on its cell surface. So now we're going to talk about that. All right, how is that going to activate? Okay, how that's going to activate our helper T cells. Okay, so here understand that most of the time, okay, I can't say all the time, but most of the time, the activation of the helper T cells occurs in a secondary lymphatic structure. So what is that? All right, well, the primary lymphatic structures are going to be red bone marrow, all right, and the thymus gland. Those are your only primary ones that we've talked about so far. Okay, secondary is everywhere else. All right, lymph nodes, spleen, all the other structures there. Okay, lymphatic vessels. Okay, so those are going to be our secondary lymphatic structures. Remember the MALT, the MALTs? You have the Peyer's patches, just these random globs of immune tissue that are scattered throughout mucosal membranes and throughout the digestive system, where we have all these immune cells just hanging out, waiting to see if something like this is going to happen. That's where this is happening. Okay, so this process is going to occur in the secondary lymphatic structure. So here's the story, all right, of a lovely lady. Okay, Brady Bunch. Come on, bear with me here. Okay, so these T cells have a specialized, this TCR is T cell receptor, okay, that will recognize that antigen. And so it'll bind to it. All right, now if we want to make it stable, all right, because it's not a really super strong, all right, connection. So in order to ensure a more stable connection, the CD4, all right, molecule, the CD4 molecule is almost like a hook. Think of it like a hook. And it's going to hook on to the MHC class II molecule. So it just stabilizes the connection. Okay, so that's what happens here. All right. So while this happens, all right, during the stabilization, it's real simple. If the CD4 helper T cell recognizes the antigen, it's going to hang around for a little while. Some stuff is going to happen. If it doesn't recognize it, it's gone. See ya, peace. Okay? Because if it doesn't recognize it, either one, it's harmless, or two, it's an antigen that it can't recognize. Because these helper cells can only recognize specific antigens. Okay? So another helper T cell will come along and look at it, okay, and evaluate it. Okay? So it's got one of two choices. If it doesn't recognize it, boom, it pops off and it leaves. And it moves on to the next APC cell. Okay? If it does, it hangs around for a little bit. Okay? So this is what we call the first signal. All right? When the helper T cell engages with, all right, the antigen presenting cell, all right, with the T cell receptor site and the MHC class II, all right, protein. Okay? So that is the first signal. So now there's a second signal that occurs. All right? So after this happens, the T cell then, all right, the helper T cell decides, I'm going to stimulate myself. So I'm going to release interleukin 2 onto myself, all right, and that will cause that T cell then to make more T cells that are specific for that antigen. Okay? Remember, safety in numbers, it's always easier to get the job done if you have more helping hands. That's what happens here. Okay? So it's going to start to proliferate and make clones that are identical to that helper T cell, meaning those helper T cells are going to have the same T cell receptors that are specific for that antigen. Good, because now those can spread out throughout the body and start looking for more of those antigens to eradicate them. Okay? So when this occurs, when that interleukin, all right, gets secreted onto itself, we've now activated that T cell. Okay? So when it starts to make more of itself, it's going to make two types. It'll either make an activated helper T cell that produces and makes more interleukin 2, and then it also makes memory helper T cells, which is cool, because now we put this into our database in case we run into this antigen a year later, then we can respond to it quicker because we have these memory T cells rolling around. Okay? The first initial encounter is always the longest. It takes a while. Okay? And we'll get into that in a little bit. All right? So that's important that you understand that. So I'm going to show you a picture here on this next page. that really, it pulls it together nicely. I love this picture here. All right, so you can see, right, here's our T cell, our helper T cell, all right, and here's the antigen presenting cell. And here you can see it's presenting all of these antigens on the MHC class II molecules. Okay? And so they can only bind with helper T cells. So they come in contact with one another. The T cell receptor recognizes the antigen, and then, all right, the CD4 molecule stabilizes that connection there. Now, there's other receptors on here that can also, it's not just one single receptor, all right, that is actually engaging with the antigen presenting cell. It could be hundreds, if not thousands. Okay, we're just showing you one. Okay, it could be just more than one, let's just say. Okay, we're just not seeing it here. So after that happens, that's the first signal. Now, that activates the helper T cell, and it starts to produce the interleukin-2 here, which stimulates that helper T cell to make clones of itself. All right, most of those clones are activated helper T cells, and what do? They produce, all right, more interleukin-2. All right, we're going to revisit that because that's going to play a role here in a few slides. That interleukin-2 just gets secreted out, okay, out and around through the body. We'll talk about that in a moment. All right, and then a smaller minority of the cells that are cloned, those are our memory T cells. And so we'll have a memory of this antigen for a rainy day. If I see it 5 years down the road, bang, I got a memory T cell, and we can start production, all right, of activated T cells. Okay, good so far? Not too bad? Cool. All right, so now that was the helper T cell, the CD4 cell. Let's talk about, all right, our CD8 cells. CD8 cells are the cytotoxic T lymphocytes. Okay, now these guys, remember the rule of 8. Okay, they'll only engage with, all right, the class 1 molecule. So where are the class 1 molecules? Class 1 molecules are in every single nucleated cell in the body. So guess what? That includes APC cells also. So if a nucleated cell gets infected and then becomes unhealthy, all right, these cytotoxic T cells can engage with those cells. They can also engage with antigen-presenting cells. Okay? So they have more choices, whereas the CD helper cells can only engage with, all right, antigen-presenting cells. Okay? Because they can't engage with the class 2 or the class 1 molecule. Okay? So APC cell or an infected nucleated cell. Okay? So the same type of scenario will happen here. Okay? So the cytotoxic T lymphocytes, all right, will engage, the T receptor will engage with the antigen, okay, that's being expressed, all right, on the class 1 MHC molecule. And then the CD8 protein will stabilize that connection. Okay, so that's the first signal. So similar to the CD4. All right. Then our second signal, remember that interleukin that I was telling you about before from that previous slide, that interleukin-2 that was kind of going off screen here? Okay, so it's going to come back onto screen with our cytotoxic T cell. Okay, so the helper T cells have been releasing that interleukin-2, and that interleukin-2 now activates and stimulates the cytotoxic T cell to do something similar that the T helper cell did. It becomes activated and will make, all right, well, let me back up. The interleukin-2 now will cause the T cells to proliferate. So they'll start making more of themselves, like we saw with the helper cells. Okay? And again, we'll see activated cytotoxic T cells and memory cytotoxic T cells. Okay, same type of thing that we saw with the CD4 helper T cells. So the memory T cells will be used for a rainy day later on if we are re-exposed to that antigen. And then those activated cytotoxic T cells are going to go around and start killing off, all right, the unhealthy or infected cells that we saw. right, which we're going to talk about in detail. All right, but for right now, we're just going to activate these cells. All right, it's like having a dog and telling that dog, go sick that person, go attack them. That's what we're doing here. Okay, our cytotoxic T cell is our dog. We're letting it off the chain. Go get them, Spike, or whatever your dog's name is. Okay, so here's a very similar scenario. So the first signal here, same thing. Okay, our CD8 cell, the cytotoxic T cell, is going to bind on to, all right, our either sick, unhealthy cell or the APC, all right, through the class 1, all right, histocompatibility complex there. All right, and then that CD8 molecule will help to stabilize that connection. And that gives time for other receptors to bind on. All right, so now off screen, you can see here's that interleukin 2. Where did that come from? That came from this T helper cell over here off the screen. All right, the CD4 cell. Okay, so it's been producing the interleukin 2, all right, this whole time. It now then stimulates our cytotoxic T cell to proliferate. It's going to make more of itself. All right, most of them will be the activated cytotoxic T cell, and some will be our memory T cells. Good so far? Not too bad, right? Okay. One more cell to go. The B lymphocyte. Okay. So here's an interesting fact. These cells, yes, they need to be activated, all right, but they can also, they're kind of like the exception to the rule. They can still respond to antigen, all right, outside of the cells that will activate them, the helper T cell. And we'll get into those examples here in a little bit, okay? So in this scenario here, don't forget, BCR, that's our B cell receptor, okay? So the B cell has a specific type of receptor. It's actually an immunoglobulin, an antibody, right? The IgD. We'll talk about that in a moment, okay? But what will happen here is, all right, the the B cell will interact with the antigen and it'll form what we call a cross-linking, all right? I'm going to show you that picture real fast. This is the cross-linking that I'm talking about. So these are the receptors, these yellow structures, and you can see here's one, here's another, and they're cross-linking. These two different receptors are sharing, all right, the same antigen. That's what we're referring to as the cross-linking there. Okay, so they're like sharing. Sharing is caring in this situation. All right, but here's the crazy part. Remember what I told you at the beginning of the class today. B cells are also antigen-presenting cells. What do antigen-presenting cells like to do? They like to play the role of Pac-Man and gobble things up, and that's what they're going to do. Okay? So they are going to bind on to the antigen, and then they're going to swallow it. And then guess what? They do the same thing that the other antigen-presenting cells are going to do. They're going to process it, and then they're going to express it on their plasma membrane so they can present it to the CD4 or the T helper cells. Okay? Like we saw with the other antigen-presenting cells. So that's going to be part of the first signal there. Okay? So same type of thing, interaction with an antigen, except in this case, they're going to go the route of an antigen-presenting cell. They're going to eat whatever they come in contact with. And then they're going to digest it up and then stick it all over their body on the plasma membrane there. And then they're going to go and communicate with the helper T cell. All right? Now, that's the first signal. The second signal is a little bit different because we are going to release a different type of chemical messenger. All right, instead of releasing interleukin 2, because we're reserving interleukin 2 for the T cells, we're going to release interleukin 4, and that's going to stimulate our B cells. Okay, so different chemicals for different cells. All right, so you can see here, I love pictures. If you haven't figured that out by now, you better. All right, so here we are. By the way, does anyone remember what naive means? It means that it hasn't come in contact yet with the antigen that it's designed to recognize. Okay? So naive means it just hasn't come in contact with whatever antigen that it can recognize at the time. Okay? So same story here, all right? It comes in contact with the antigen, we cross-link it, all right, and then it ingests the antigen, packages it up, okay, and then expresses it onto the cell surface here, like any good antigen-presenting cell will do, all right, onto the MHC class II molecules. And then our helper T cell will come rolling along here, all right, check the antigen, make that connection there. It becomes activated because it recognizes the antigen, and then it starts to produce the interleukin-4 here onto our B cell, okay? And now we've activated our B cell, and our B cell now can do what it likes to do. It likes to make antibodies, amongst other things, okay? So now the B cell has a choice, okay? Well, before it does anything, it's going to, once it's activated, proliferate. Remember, safety in numbers. Let's make more, you know, many hands decreases the work. So it starts to proliferate and make more activated B lymphocytes. Some of those activated B lymphocytes will turn into memory B lymphocytes. Again, you know what those do. Rainy day, we can make, all right, more antibodies at a later time. Most will undergo what we call differentiation, and they'll transform into plasma cells. And it's the plasma cells that make the antibodies. Remember that. All right, B cells don't make antibodies. Plasma cells do. The plasma cells come from the B cells, the B lymphocytes. Okay? So once we activate our B lymphocyte, all right, it's going to clone itself. It's going to make a lot of copies of itself. Some of those clones will be memory B cells. Most will become plasma cells, and the plasma cells make antibodies. And so today, we're going to talk about all the stuff that antibodies do. You're going to become so sick and tired of antibodies. Okay? Okay, so now we've activated our B lymphocyte. All right, so it's going to make clones, so it's going to reproduce or proliferate, and then it's going, I should say, will differentiate or turn into our plasma cells, and they make the antibodies. Okay, the remainder will turn into memory B cells. Okay, interesting fact, memory B cells live longer than the plasma cells. That's okay though, because if you get re-exposed to that same antigen, your memory B cells, when they get activated, guess what? They're going to do the same thing. They're going to proliferate, all right, and then they'll differentiate and make new plasma cells. That's cool. So we don't need these old plasma cells lying around. That's fine, all right? It's nice to have them, so it doesn't take as long, but, you know, we're fine, okay? So this is, remember I was telling you, all right, we don't always need activation with those T cells, okay? So we don't need to, but the problem is, all right, if we are going to make our memory B cells, all right, we need T cells to be present. Okay. So if you've ever watched a TV show, and if you're into the rom-coms or whatever, there's the same old storyline, especially around Valentine's Day in some of these movies, where you have somebody that was broken up with their boyfriend or girlfriend, and they're sitting at home, and their friends are like, come on, you got to go out. You got to go out. You can't sit here and feel sorry for yourself, okay? And so the person's like, no, I'll just stay at home. I'll meet somebody. And their friend says, listen, you can't go out. If you don't go out, you're not going to meet anybody. All right, you have to leave your house or apartment and go out to meet somebody. Well, guess what? All right, that same thing is true, all right, for our lymphocytes. If our lymphocytes don't go out every once in a while and circulate throughout the body, they're not going to meet that one special antigen that they were made for. All right, it's like the true love of lymphocytes. Okay, so that's what happens here when we're talking about lymphocyte recirculation. Okay, remember, most of our lymphocytes aren't in circulation, not in the blood or the lymph. They're mainly in tissues. All right, secondary lymphatic tissues. So every once in a while, they're going to go out and circulate. Okay, they're going to mingle amongst the common folk. All right, and they're going to go out through the blood, the lymph for a couple of days. All right, And this gives them more of an opportunity to find that special romantic encounter with that antigen that they were made for. And when that happens, then it's party time. Okay? Because we're going to activate some lymphocytes and have ourselves some fun. Okay? Okay, so now we know how to activate these cells. So now we get to learn how what their effect is going to be. Like, how do they win? You know, your body gets smashed with all these virus particles or bacteria particles. All right, how are we going to stop, all right, this antigen, this foreign molecule, and all of its clones? How do we kill all this virus? All right, how do we kill this bacteria or these parasites or these fungus molecules? Now we're going to talk about the effector response, okay? And it varies depending on which type of cell we're talking about, all right? So you have the three types of cell, all right? Our CD4, okay, or the helper T cells, our CD8, all right, the cytotoxic, and then our, well, B cells, but plasma cells, okay? So the helper T cells, this is what, and if you notice, each of these cells, all right, has a kind of a different effect, okay? See, the helper T cells don't actually go in and do the dirty work themselves. See, they're the mastermind. They get other cells to do the dirty work for them, okay? So how do they do that? They release chemicals, interleukin-2, interleukin-4, all right? They like to release chemicals, and those chemicals tell these other cells, all right, that are from the innate immune system or the adaptive immune system, okay? If we're talking about the innate, we're talking about macrophages. We're talking about the natural killer cells. That's the attack dog. It just runs around and kills things, all right? So they'll influence the natural killer cells, amongst other cells, to go and do the work, all right? Whereas the cytotoxic T cells, they go in and do the dirty work themselves. How do they do it? They're going to go and destroy those unhealthy cells, and we'll get into the specifics, through apoptosis. They're literally going to drill holes into the plasma membrane. Remember the perforins and the granzymes, right, that the natural killer cells use? Well, these cells use the same type of scenario. They're going to stick holes in the plasma membrane and then pump in these digestive enzymes that are going to just destroy the inside of these unhealthy cells, and these unhealthy cells will die via apoptosis. And then finally, our plasma cells are going to make antibodies. And we're going to talk about all the different ways that antibodies, all right, are going to end up being a part of the immune response. Okay? All right, so let's go into more detail now about specifically, and I've talked about this, but now I'm going to kind of pull it together. Let's draw it together so it's a little bit more understandable. All right, when we're going to talk about the effector response for the helper T cells, CD4 cells, CD4. Okay? So at this point, we've activated. They've been exposed to the antigen. They're now activated, which means we have activated T helper cells, and we've made some memory T helper cells. Okay? So in this scenario, let's go way back to 2 classes ago. Remember that picture I showed you of the kid on the skateboard that wiped out and scraped his knee? Let's use that scenario, okay? So same story, kid scraped his knee, some bacteria managed to get into the wound, okay? Some of that bacteria managed to make it into circulation, and it got flushed around, all right, your blood system, your lymphatic system, all right? And these antigens finally found their way to our T helper cells. So now they're like, okay, guess what? You know, we've found that there's a problem somewhere in the body, and we have to go to the source. It's like watching, I don't know where I get these analogies from, but they just pop into my head. And there are a lot of them are from movies or TV shows. It's like when you're watching a horror movie, and these people are sitting around a campfire in the middle of the woods, and there's a serial killer out there. They're sitting around the fire, and they have a dog, and the dog runs off into the woods, and then it comes back carrying a hand. All right? And so they're like, where did this hand come from? Let's go find out where the body is, all right? And they go off to where the dog found the hand. Well, that's what's going on here. All right. So you want to think of, all right, the CD4 cell, all right, it found the hand somewhere, all right, or that the hand floated through the circulation, all right. The CD4 cell, the helper T cell found it, and now it's like, all right, let's go to where the infection site is. So when it gets there, right, the CD4 helper cell, or T cell, is going to release all these different types of chemicals, the cytokines, okay? Those chemotaxic agents, or chemo-attracting agents. And it's like shooting a flare-up if you're abandoned somewhere in the woods. Now we can find out where you are. Because now we want to get more cells. The T helper cell does not do the heavy lifting. It gets other cells to do it. But it's going to be the guy that's going to be like, hey, I'm yelling, come on over. We're going to get more immune cells to come in. So they go to the site of the infection, and they send out all their chemicals that are going to attract other immune cells to the area. Neutrophils, all right, macrophages, natural killer cells, all of them. Let's get them all in here, all right? We got all this bacteria that's running around this infection site, and we got to kill it, okay? Also, this is supposed to say helper, by the way, okay? They're going to activate the B lymphocytes. How? Through the release of cytokines, interleukin-4, okay? That'll activate the B lymphocytes. When that happens, then the B lymphocytes can then proliferate and differentiate into memory B cells and plasma cells and make antibodies. Okay, because maybe we want to kill that bacteria off with some antibodies. We can do that. All right, guess what? They're also going to activate our CD8 cells, the cytotoxic T cells. Same thing, cytokines. All right, what was the one that we were talking about before? Interleukin-2. All right, interleukin-2. And we talked earlier on earlier on in the immune system about how it's going to get some of the innate cells, the macrophages, all right, to help out. Okay, it's going to stimulate it, all right, same type of thing through cytokine release. Okay, so you can kind of see the helper T cell does a lot. Okay, basically finds the site for the infection, it gets there, and then it just starts releasing all these chemicals into your circulation. Goes throughout the body, all right, and that's going to attract these other immune cells to the area of infection, and then they can do the work on killing off the bacteria, the viruses, whatever the infectious pathogen is. Okay, so they're the signaler. All right, what about the CD8, the cytotoxic T cells? How are they going to handle it? Same type of scenario, all right? Exposed to an antigen, we've now activated the cytotoxic T cells, all right, and they have proliferated into more activated cytotoxic T cells and memory T cells. Where do they go? To the infection site. They're going to the party, okay? And so now, in this case, they're not going to get other cells to do their job. No. They're going to look around at any of the cells that are expressing, all right, the foreign antigen, okay? And if they are, they're dead. We're killing you. Okay? So any of these cells that are expressing, all right, via the MHC1, all right, class 1 and class 2, all right, molecules, all right, they'll go through and start slaughtering them. As long as they're expressing, all right, that foreign antigen there. All right? So how do they do it? Same way as the natural killer cells. First, they release perforin that starts perforating and punching holes inside or into the plasma membrane, and then it pumps the granzymes into that hole, all right, and it just starts killing the inside of the cell. And that cell dies by apoptosis. See how we're kind of drawing it together now? I hope, all right, kind of helping the story out a little bit, all right, un poquito. So that's what we're going to see here now. We've activated our T cell, the cytotoxic T cell. It goes to the area of infection, all right, and it just walks around. I shouldn't say walks around, but it moves around that tissue, that area there, and it looks for any unhealthy cell. And it knows it's unhealthy because it's expressing that foreign antigen. And then it just starts punching holes into these unhealthy cells and killing them with the granzymes through apoptosis. That's it. Okay? All right. So that's the effector response, and I'll show you a nice picture of that right here. Okay? So here is our activated cytotoxic T cell. All right? It's connecting to the unhealthy cell here. It's pouring the perforins into the plasma membrane, making the holes, and then the granzymes just enter in and just start destroying the inside of the cell. And it causes that cell, right, to undergo apoptosis and die. Simple. Simple, simple. Okay? All right, the last effector response is for our B lymphocytes. Their effector response is to become another type of cell altogether, the plasma cell. And it's the plasma cell that makes and releases the antibodies into the circulation. Now, the interesting thing is they do this from afar. They're like the general, all right? Not of this whole process, but the general's not on the front lines. No, the general is at command center, all right? So the B cells, the command center, is going to be in the lymph nodes, all right? I shouldn't say the B cell, excuse me. Well, there's B cells in there too, all right? But the plasma cells command center are going to be in the lymph nodes. And so it's making the antibodies, it releases the antibodies, all right, into the lymphatic fluid, which eventually make it into circulation, which will eventually make it to the infection site, all right? So the nice thing is about these antibodies is that they'll stick around, all right, and they'll keep producing, or see, the B cells, or excuse me, the plasma cells will keep producing for 5 days. That's a good long time, right? And it's literally producing millions of antibodies. Million. That's just one plasma cell. Okay? So they're going to wander around through the circulation of the lymphatic system, of the blood system, and go through the tissue. All right? And they'll wind up, all right, hopefully at the infection site there and start attacking. We're going to go through the different ways that the antibodies function, all right, because there's about 6 different ways that antibodies can handle, all right, an infectious agent. And we're going to talk about those. So if you ever go, like when people talk about, I'm going to get an antibody test for the coronavirus, what they're doing is they're checking the antibody titers. That's what they're doing, okay? It's basically just checking, all right, how much of that specific antibody to the coronavirus do you have in circulation, okay? So we call that an antibody titer. I got to take a sip.
Have you had anything? Have you had one now?
No, I'd like to, but I mean, I had mine back in December. So if it's true about the 3-month thing, I probably might not have much, if any, but I've read some research articles out of Australia and I think France, in which they were saying that the antibodies can stick around anywhere between 5 to 8 months. So there's a possibility that it would still happen. I tell folks I had a positive COVID test. I didn't really have the traditional symptoms. I think I had COVID, you know, but I mean, if I don't have the antibodies, then I'm going to start thinking that less that I did. Because there's no way that you can have something like that and not mount an antibody defense.
Yeah.
Okay, so what the heck is an antibody? All right, it's an immunoglobulin. Remember those proteins that float around in the blood that we were talking about? Part of the 7% of the proteins that linger in your blood plasma. Okay, the immunoglobulins are part of the 7%. Okay, and they are made to fight a specific or particular antigen. Now the article, and this is I can't promise you this is the last time I'll mention that article. But that article that I sent you guys, they talk about how the antibodies, and I'll get into more specifics of it, how they're able to refine their production and make what we call the variable region, all right, of the antibody more specific and more capable of handling infection. I'll explain that to you in a moment. But basically what they do is they'll tag, all right, those antigens, all right, and signal, all right, that hey, this is a thing that needs to be destroyed, and then the other immune cells will come over and handle that. Okay. So as you can see here, viruses, bacteria, toxin, yeast spores, all right, even parasites, all right, these antibodies are great against them. You notice the first thing on the list is viruses, because they work wonderful in that way. All right. So what we'll see is, especially if, what does soluble mean? Do y'all know what soluble means? I wasn't 100% sure of what it meant until my daughter defined it for me earlier this school year, so I needed a nice refresher. It means that you can dissolve something into a fluid, essentially, all right? It can dissolve, something will dissolve into a solution, okay? All right, so these soluble antigens that can dissolve into a fluid make them harder to detect if they're soluble, okay? We want to bring attention to these antigens. We don't want them to go unnoticed. So we want to make them either bigger or we want to make them insoluble. All right, the humoral immunity has to do with the B lymphocytes, by the way. Cell-mediated immunity has to do with the T lymphocytes. Both cell-mediated immunity and humoral immunity are a part of the adaptive immunity, what we've been talking about the last two classes. Okay, so most of the antibodies are going to be in a Y shape. And they too are soluble. Well, that's cool. So they can dissolve into the blood plasma and just float around. I like that. All right, I got to go through this part here. This is a quick anatomical description of what an antibody looks like. Okay. So you have two identical heavy chains and two identical light chains. This is what the light chains are and the heavy chain. This right here, this yellowish structure, that's the heavy chain. And here, this little kind of whitish area, that's the light chain. Okay? Both of those make up what we call the constant region. That doesn't change. Okay? Where am I? Did I skip? Okay. So you'll also notice, okay, that in between, all right, in that Y section there, we have what's known as the hinge region, which is this region right in here. Okay, that just allows, all right, the Y part to be more flexible, to move around, so it can attach to an antigen easier. Okay, it helps that out. All right. Now, if you look here, there's two sets here. All right. There's a part over here and another part over here. Well, we have to hold those together through this disulfide bond. That's important to know that, because anything that can digest a sulfuric bond can destroy this and destroy our antibodies. And that's no good. We don't like that, okay? And then finally, at the bottom here, the two functional areas that we will discuss, I'll talk more about the constant area, all right, are the variable and constant regions here. What are those? Well, you saw me talk about the constant region. That's pretty much all this area. And at the very end, we have the variable region. And this is the part, all right, that undergoes constant refinement during the immune response. So by the end of an immune response to a specific antigen, the antibodies that are made are awesome. In the beginning, they're good. They can get the job done. But at the end, by the time the infection is pretty much over, all right, this variable region, all right, because we're basically plug and playing. We're just trying, does this work okay? All right, this works okay, but what about this and this? And so we start to change around the genetic structure here of the variable region, only to make it better to bind on to the antigens. All right, so that's why that area is pretty important. Okay, so at the variable regions here, at the ends there, we refer to those as the arms. And that's where we're going to bind our antigen here. Okay, and when we bind the antigen, they're relatively weak binding interactions. All right, which is good, because if it's strong, then it's going to be hard to pop that thing off of there. Right, so but you don't want it to be too weak, and it's not, because if you look, hydrogen bonds, all right, those are relatively weak. Hydrophobic aren't too bad. Ionic bonds are a little bit stronger than the hydrogen bonds there. But those are all considered to be relatively weak, all right, types of bonds, which is okay. It's not a bad thing to have that happen. Now, our constant region, all right, we call that the FC region there. All right, this is how we determine what type of function in the body, what type of reaction that the actual antibody is going to undergo. And I'm going to talk about those functions here in a second. All right, you should know these classes of antibodies. All right, The nice thing is the constant region is the same, okay, throughout the class. So all the antibodies in a class D, all right, antibody class are the same. All the constant regions in IgA are going to be the same. Okay? So those are the 5 major classes listed below: IgG, IgM, IgA, IgD, and IgE. All right, so we're going to talk about each of those individual classes here in a moment, but first I want to talk about, all right, some of the actions here, okay, of the antibodies. Now there's several. So what I'm going to do is, and some of these you've already seen before, and we talked about the complement system, okay, so it's going to be a slight review. Okay, so let's talk about the first one, neutralization. Where did I hear that before? Maybe in chapter 2 when we were talking about acids and bases. You have something that's too basic, all right, we add acid to it, all right, we try to neutralize it, okay? This one here, all right, is going to be when we're dealing with, remember the antigenic determinant? You might not remember it. I don't know if you remember the term epitope. All right, I'm going to go back here while you're writing something down here. Let me see if I can find that one slide there. No, that's not it. All right, an epitope, or the antigenic determinant, come on, where are you? There we go. I'm going to come right back to this slide, slide 81. That's this thing. Okay, so here's our antigen. All right, it's either our foreign protein or polysaccharide. See these little nubs that are coming off? Okay, those are the antigenic determinants. Those are the things that are going to grab on to a cell. All right, so this antigen, if it's a virus, can then enter into the cell. Okay, so think of it like as an arm. Okay, and it's going to grab onto a cell receptor. Well, you know, hey, what's a good way to make sure that this thing can't bind onto a cell? Let's chop off all its arms. We're not going to chop off all its arms, but we're going to cover up all its arms. So that's what we do with neutralization. All right, we're literally, all right, the antibodies are going to cover up all these antigenic determinants. And that way they can't connect to another cell. All right, so they can't bind to a cell receptor. So we just covered them up. It's a pylon. Here, let me show you the picture. That's what we're seeing here in this picture. Okay? Now you can't see it, all right, but we would assume that these blue balls here, all right, those are the epitopes, all right, or the antigenic determinants here. Okay? And so these antibodies are just covering up all those areas so they can't bind onto anything. That's neutralization. Okay? Second one we saw before in blood. Remember we were talking about the ABO blood classes and whatnot? Okay? And so we saw what happened in the blood. Similar here, we're going to get cross-linkage, all right, with the antigens, and it forms clumping. Now remember what I said before. All right? Ideally, antigens want to go under the radar, unnoticed. You know, we don't want to be seen. Okay? So do you think it's easier, all right, if a person that is 3'5" is sneaking around in the middle of the night, all right, wearing all black, and it's dark out? Do you think it's harder to see them compared to a person that is 500 pounds, 6'7", and wearing red in the middle of the night? Okay, so what we're going to do here with agglutination here is we're going to create these cross linkages, and we're going to start causing clumping. Well, guess what? We've got cells that are constantly moving through everywhere. When they see that clumping, that irregular formation, they're going to go investigate it. All right, cool. If it's a macrophage, or it's some type of phagocytic cell or a natural killer cell, because we know that those types of cells are non-specific. If they identify something that they don't recognize, they're going to go and kill it. Yo, you did. All right, Morgan, thank you. I didn't even see that. Did you just send that a while ago? You did read it. Good, good. Well, recommend it to everybody else you know, Morgan. Your classmates, specifically. It's good. All right. Okay, so that's agglutination, okay? And it works great, 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 all right, when we're talking about bacterial cells. So that's what we're seeing here, okay? So you can see, all all right, these antibodies are going to create a cross-linkage between two bacteria here, and they're going to start to create these clumps. That's going to draw attention to them. And then so one of our immune surveillance cells or our non-specific innate immunity cells will come by and see that, and they're like, oh, the heck with that, killing it, okay? All right, the third one here is precipitation. All right, similar type of situation to agglutination. In this case, though, all right, we're going to have another cross-link occurring. All right, we'll see it with the viruses, but now we are going to make, remember what I said, most of these antigens are going to be soluble. They'll dissolve right into the fluid. Well, when precipitation occurs, now the antigens, all right, cause those, the antibodies now cause the antigens to become insoluble, and they precipitate out of the body fluids. Is anyone here familiar with Star Trek? All right. Well, in Star Trek, there's a race in there called the Klingons, and the Klingons are this combative race, and they have this technology called cloaking. All right. And so they can basically turn their ship invisible. So think of it like that. All right. When something's soluble, it turns invisible. You can't see it in the fluid. But when it becomes insoluble, now it turns off the cloaking device, and you can now see it. And that's what happens. When it becomes insoluble, it's visible, and now the other immune cells, all right, can come along like a phagocytic cell and gobble it up. All right? And that's what we see here, all right, with precipitation. Okay, and that's what we're looking at here. Okay, so here are all your little virus particles wandering around. And then your antibodies now make another cross-linkage type of complex here. All right, and it keeps getting bigger and bigger as long as we have antibodies and antigens. And that causes, all right, all of these substances to precipitate out of the fluid, and basically we're turning off the cloaking device, and we can now visibly see, all right, these molecules, and then our phagocytic cells, all right, antigen-presenting cell, all right, macrophage, all right, can gobble it up. Cool so far? All right. The last three types of functions for antibodies. Complement. Well, I remember complement. All right. That's that part of the innate immune system that can cause a couple different things. It can cause inflammation, opsonization. All right. It can also cause some clumping going on. Well, here, all right, we're going to see now with these last three, all right, functions all have to do with the FC region, all right, the constant region of the antibody. What's that? Has to do with this part down here, okay? The bottom of the Y, okay? So keep that in mind. So now we're going to talk about the bottom of the Y, okay? So we talk about IgG and IgMs. One, IgG is the most abundant type of antibody in circulation there, okay? So what it will do is, okay, it will, the antibody will bind on to the antigen, and then that antibody will also bind on to the complement. And then we can undergo that classical complement activation. Well, what the heck is that, Dr. Kaz? Well, if we go all the way up to the top here, I'm not going to go through all the different parts of complement, all right? But I just want to show you the one that is the most common that you'll see, all right, is this one right here. Elimination of the immune complex, okay? We can have a couple things. We're going to talk about opsonization here in a second. But what we're talking about is this type of scenario. All right, now that the antigen is bound onto the antibody, then it binds onto the complement. That complement can jump onto the red blood cell, and it can go to the spleen or the liver, and it can get cleaved off, and it can be destroyed by immune cells in those. Well, we know that the spleen has plenty of immune cells in there, all right, and so does the liver. All right, the liver has plenty of macrophages that like to live in there, and it presents it to the macrophage, and it gets killed. Okay. So that's what we're talking about when we're getting that the activation of the classical complement pathway there. Opsonization here, okay? Again, we're going to be dealing with the constant region here. All right, but now what we're going to do is you're going to see, all right, well, let me just show the picture. I hate trying to read the words. All right, So here is your antigen. Now the antibody, all right, jumps on, where am I going? Here we go. And the antibody jumps onto the antigen here, and then the constant, the FC region, then can make contact with the macrophage, which again is an antigen-presenting cell. All right, so it's going to investigate it, but the antibody now activates the macrophage, and it doesn't have to go through all these other steps, all right, to get the macrophage to do what it does best, and that's going to be to phagocytize, all right, this this antigen here. Okay, so that's just a direct way, all right. So we don't have to release this interleukin to do this and that, all right. But basically, what it does is the antibody skips all that stuff. Okay, and it directly activates the macrophage, all right, to gobble up the antigen. Okay, and then the last one here is natural killer cells. Same type of scenario. Okay, the antigen is floating around, all right, the virus is floating around, or the bacteria is floating around. The antibody binds onto it, all right, by the variable region, all right. Then the constant region, the FC region, then is going to then cause an interaction between the natural killer cell. Then that natural killer cell is going to release the perforins and the granzymes, all right, and destroy and rip open, all right, that antigen. That's what we see here. Okay, so here is the infected cell or whatever antigen. All right, the antibodies have bound onto it. The FC region will then directly stimulate the natural killer cell to destroy this cell here and causing it to undergo apoptosis. All right, so you can see that it does quite a lot of stuff, all right, these antibodies here. All right, questions about that so far? I'm actually moving through this much faster than I thought I would. Hoorah! Good job, y'all. All right, so there's 5 classes of antibodies, okay? And they're listed above here. We're going to run through each of these relatively quickly. Okay, the most abundant type of antibody is the IgG. All right, this is also the one that can cross over the placenta, okay, which could be bad, especially if we're dealing with a situation in which mommy's blood type is different than baby's blood type. All right, these antibodies, all right, when they cross over the placenta, can cause that hemolytic disease in which it can destroy, all right, the red blood cells of the newborn. All right, that's where we were talking about, all right, I was going to say the RH factor, okay? If you're positive or negative, that's where ROGAM comes into play, okay? All right, the next type is the first type of antibody that's usually formed. Okay, the IgM. Now this is the one that does not look like a Y. It actually has 5, if we were to take, I can't draw them all. If we were to take 5 Ys and stick them all together at a common center point, that's what it looks like. Okay, so that's what we refer to, whoops, that looks awful. Okay, all right, but it has that pentamer structure. That's what we're referring to. This is the first type of antibody that usually gets produced. All right, so if you think about it, all right, it has all, look at all these variable region binding sites for antigens. There's a ton of them. So it's great for things in which we need to do cross linkages. So that would be agglutination, all right, and binding complement. Okay, awesome job there. All right, we'll also see these in high numbers, all right, when we're dealing with transfusions, blood transfusions. Okay, if you have an issue, if you've mismatched, it could be a problem. Okay. All right, the next type of antibody, IgA. All right, IgA, and I talked about this when we were talking about the mucous membrane. This antibody is made in abundance in mucous membranes. Well, where do we find our mucous membranes? In areas that are going to be close, all right, to the external environment. We were talking about the respiratory system. All right, the mucous membrane of your nasal mucosa, of your pharynx. Okay, of your throat, of your respiratory system. Okay, so anything that's going to be exposed to the external environment, mucus, saliva, tears, breast milk, all of those. Okay, we'll see it in the respiratory tract. We'll also see it in the GI tract. Okay, So these guys are great for agglutination. IgD, this is the one I was telling you about that will act as a B lymphocyte receptor. Okay, so when we see that these B lymphocytes are expressing, all right, these types of antibodies on its surface, then we know it's ready for activation. Okay, so that's how we know. That's easy. And then one of my favorites, because I have to deal with this one all the time, is IgE, all right? Allergic reactions, okay? So you're going to see it in allergic reactions and with parasites. Well, there's some cells that we saw that also dealt with allergic reactions and parasites. Basophils and mast cells, all right, for the allergic reaction portion. Eosinophils for our parasite infections, okay? So usually, normally, if you want to be able to tell if somebody's having an allergic reaction or if there's some sort of chronic allergic reaction that might be going on with them, you do antibody titer on them, and if they have elevated IgE, then you can actually then start to investigate possible allergens for that person there. All right, so for the most part, the plasma cells are going to be releasing a specific type of antibody, okay? But then there comes a time when they need to stop making a certain type of antibody and switch to make another type. We call that class switching, all right? But we need the help of, guess what? The helper T cell. And it's when the helper T cell, all right, will then stimulate, all right, that plasma cell to switch over, and it's dependent on the type of chemical or cytokine that's released, which will tell that plasma cell, okay, stop making the IgAs. Now you can start making IgEs. We don't need that anymore, okay? But it takes the helper T cell, all right, to do that, to help stimulate that. All right, questions? A lot of that stuff that we just covered is just straight up memorization. I hate to say it. Okay? All right, so let's get into something that's a little bit more practical for us as normal humans living in America, okay? All right, because you hear a lot about vaccinations and things of that nature. So what's the purpose of a vaccination? I know it's supposed to help me, but how? Why? All right, well, why is obvious. I don't want to get sick and die, all right? But how does it help me? So we're going to talk now about the immunologic memory, okay? So now we've activated B cells, we've activated T cells, and they've made their memory, quote-unquote, cells. All right, so what good are they? Well, again, if you're re-exposed to that antigen, then we need a way, all right, to form a long-lived army, all right, of lymphocytes for another invasion, okay, because it's war. It really is. It is a form of war, okay?
So why can't you do that with like allergies?
Well, we're going to talk about that, the hypersensitivities. Yeah, I know. And, you know, in some cases you can't. Here's the problem. It's not, like, allergic reactions aren't the fact that your immune system isn't working. It's working too much. It's overdoing things. All right, it's producing too much, or it's working when it shouldn't be working. All right. Okay. So again, when we're talking about, all right, immunologic memory, okay, we're going to be dealing with the adaptive immunity. Remember, your immune system has two parts to it. Innate, which is like the preventative defense, okay? And then the second part, or the other half, is adaptive immunity. And adaptive immunity is all based on an antigen and the lymphocyte and the interaction that happens between them. That's it, okay? So it involves an antigen and a lymphocyte, and then what happens with that relationship. Okay, so we're going to talk about, all right, now we know that the very first time that you're exposed to this specific antigen, it takes a while. That's why you get the flu shot, okay? The flu shot is just a guesstimation of, okay, these strains were prevalent in the past year, so in case you meet up with it the following year, all right, there's a high likelihood you'll meet up with one of these strains, okay? And if you do, awesome. Because now our vaccination has already exposed you to the antigen, So when you're re-exposed to that antigen through going outside and being close to somebody that had that flu strain, all right, then your response time, all right, to it will be much shorter. You'll be able to fight it out. And you might not even know that you were exposed to it because re-exposure could be so short because your body mounted such an awesome response to that, immune response to that re-exposure. You might never know. You might never be symptomatic. Hopefully, okay? So when we have a first exposure, all right, we're going to talk about what the lag time is, the time that it takes for your immune system to start to kick in, all right, for things to start working, okay? All right, so keep in mind now, all right, when we start getting into this, all right, when we talk about activation, all right, because when you activate the immune system, you're going to be then activating memory cells if you have them for that specific antigen. If not, you're going to make them, okay? So we're going to actually start to produce and create memory cells, all right, because you're not going to always carry a full load. Over time, they just start to die off, okay? Okay, so when we talk about a subsequent antigen exposure, that is just saying when you get re-exposed, you get exposed to that antigen again, and maybe again, and maybe again, all right? So if you have memory cells for that antigen, well, they're going to actually then be able to mount an immune response in quicker time, all right, compared to the first time. So we refer to that as the secondary response. You have a primary response and a secondary response. So the whole point of giving vaccinations is for that. Developing memory. We're going to develop memory because, again, exactly when you take a vaccine, you're just giving that person either a dead form of the antigen or a weakened form, if it's a virus or a bacteria. Oh, I have not researched what's in the COVID.
That's what the document says.
That there's no COVID actual antigen in the COVID. Oh, I'll have to check because I know there's the spike protein on the capsid. Yeah, you'll have to show me. I'll be curious about that.
You know, people are like, "Oh, you're gonna get COVID." I was like, "Well, this is not in there."
Oh, I haven't done that much research on it only because there's just so much stuff out there.
Yeah.
Okay, so let's talk about what happens now, all right, with a primary response, which is pretty much what we've been talking about, and the secondary response. I'm going to graph both of them for you here. All right, so you can see here's the primary, the very first time that you're exposed to an antigen. Here's the secondary response. So if you notice here, all right, this light blue is the lag time. And the lag time, look at that. It looks like it's 3 or 4 days. All right, that's how long it takes for you to start to mount an immune response. And then when you do, okay, here are the levels of your antibodies, the IgM and the IgGs. All right, they're not too high here. Okay, and look how long it takes. All right, from the very first day of exposure. All right, so look at this, 14 days is more at peak antigen or antibody production. It's 2 weeks. That's garbage, all right? Now look at the secondary response, okay? You have a much shorter lag phase, all right? But now you can see within the first couple of days, bang, IgG takes off. And in fact, it's made more and first before IgM, okay? So you can see the difference. This is ideal. This is what we want. This is what happens, hopefully, with me the second time or third time or fourth time when I'm exposed to coronavirus, all right, because I have those memory cells floating in my body, all right? My immune response should be like this. The very first time I was exposed to it, my immune response was like that, okay? Not a lot of antibodies, okay? So when we talk about the primary response, our lag phase can be as long as 6 days. And so in that period of time, we can't detect antibodies. All right? So the primary response could be one of 2 ways. It could be direct contact, where you get the antigen naturally, or from a vaccination. Okay? So when that happens, and everybody experiences this with the vaccination, all right, especially if you've never had the antigen, okay? So this is how long it takes. So what happens during the lag phase? Check it out, all right? It takes, during that time, all right, a little bit, during the lag phase, a little bit of time to find the antigen and not only find it, but detect it. Then we have to activate our lymphocytes. Then our lymphocytes need to clone and make more of themselves. And then if we're dealing with B lymphocytes, which we are, because we're talking about antibodies, then we have to change those B lymphocytes into plasma cells to make antibodies. All that happens during the lag phase. That's a lot. It's a lot of stuff to happen. That's why it takes so long. Okay? And now we have now we have the plasma cells producing antibodies. That's going to take up to 1 to 2 weeks. We start off with the IgMs, and then the IgGs take over in production. Okay? So this could take as long as 14 days to mount a decent immune response. Okay? For the primary exposure. That's the primary exposure. All right, secondary is much better. Okay, so you get exposed to this a second time or a third time. All right, first of all right, we decrease the length of time that you have to undergo, all right, antigen detection, activation of T cells, or activation of B cells, and then to proliferate the B cells, and then to differentiate the B cells into plasma cells, and then to produce the antibodies. All right, that's way shorter. Way shorter. Why? Memory cells. Those memory cells are floating out there, and so we don't have to go through all that now because we have them present. We just activate those memory cells. Those memory cells, we're just cutting out a couple steps. What happens with those memory cells? Right? They're going to proliferate, and then the majority of them are going to differentiate into plasma cells and then start producing antibodies. Okay, so this happens very quickly. As you saw on that graph there, all right, look at it. You can see here, all right, within a week, your antibody levels are skyrocketing. They're huge, huge. Perfect time to take a break. Wow, I did not expect to make it this far. You guys are rocking. All right, so I'll let y'all finish writing that stuff down. Let's take a break. I'm going to finish this up in the lab section. Okay, there's only 6 more slides, and then we'll do the respiratory system. And we could be right on target. Okay, so let's take a break.